Here's your introduction to the urinalysis lab, which will be our last lab that we're doing from the lab manual. And so let's, as usual, go over the important terms. We're going to start out with micturition, and that is a fancy term for peeing, sort of the medical term for peeing. Now, when it comes to physiology of the kidneys, you really have to understand the functional unit, and that is the nephron. And at the nephron, the following three major processes take place: filtration, reabsorption, and secretion. So I'm going to draw a nephron, and then we're going to talk more about where each one of these three major processes occur. Okay, so here's my version of a nephron. That's the functional unit in the kidney. You have about 1 million nephrons per kidney, and these important processes of filtration, reabsorption, secretion take place here. They're labeled here with abbreviation. So G stands for glomerulus. This is where we start. So blood enters the glomerulus coming in through the afferent arteriole, and then we have a very leaky capillary bed here. And tons of stuff, mostly water and many other things will be filtered out. Anything that's small enough to leave will be filtered out. So lots of water, glucose, nutrients, a lot of things. And collectively, all your 2 million nephrons, they will filter about 180 liters of fluid per day. So 180 liters will be filtered per day. That's a huge amount of fluid. Now, obviously, you can't lose that much fluid, so you need to reabsorb most of it. And so the job of reabsorption is a major one, and starts right after we're done here with filtration. So filtration, that's your 180 liters. Then reabsorption needs to take care of 178.5 approximately liters. So we need to reabsorb the vast majority of what we filtered, and that's the job of the proximal convoluted tubule, the loop of Henle, and the distal convoluted tubule. Once we're reaching the collecting duct, we're pretty much on our way to the bladder, and there's not much happening there anymore in terms of reabsorption or secretion. So here we're to the bladder. Now, when you listen to a lecture, there's so much more detail in how the proximal and the distal convoluted tubule and the loop of Henle, how they are reabsorbing fluids. And so you should definitely go over this. But here for the lab purposes, we're just going to go over the basic processes. The glomerular filtration rate is the per minute volume of fluids that are being filtered at the glomeruli. And we are talking here collectively about these 180 liters per day. And so each, you can divide it, I guess, by 2 million, and then you would find out how much each glomerulus actually filters. But anyway, so the glomerular filtration rate puts then a, time increment to it. So it's usually measured in per day or per minute. So that's the amount of fluid filtered at the glomeruli collectively. Now we have one more term up here, the secretion. Secretion is a small inward force that puts stuff back into the collecting duct here in the distal convoluted tubule mostly. So secretion is a small amount of material that your body decided sort of at the last minute to get rid of. And it's mostly like hydrogen ions and some other stuff, some toxins maybe that your body wanted to get rid of. So that would be secretion. It's a very small amount. Now let's move on to more lab-specific kind of terms. When you do a urinalysis, what we would have done in the lab is put a urinalysis dipstick into your own urine sample that you would have collected, and then you measure different things. Standard practice would be to take a look at the specific gravity, take a look at whether there's glucose present or proteins, and so those things. So basically for the rest here, the majority of the time we're going to take a look at whether it's normal to find certain substances in the urine or not. So specific gravity is a measure for the density of a liquid, and it means the higher the specific gravity is, the more stuff is dissolved in this case, your urine sample. So if you have very highly concentrated urine, that means you will probably kind of dehydrated, you don't drink a whole lot of water, then there's more stuff dissolved in the urine sample, and that means the specific gravity is higher. So that's just a number that gives an indication of how concentrated the urine is and sort of how well hydrated you are. And diabetes is a condition where you don't have enough insulin or maybe no insulin at all, and so therefore you cannot take glucose into cells, and so that means the glucose accumulates in the blood and you have a blood glucose level that's way too high. Now, glucose is always filtered out here at the level of the glomerulus, so all the glucose will leave and go into with the fluid into the proximal convoluted tubule, but then you have lots and lots of glucose transporters that will reabsorb the glucose, and normally these reabsorption mechanisms, they can handle a normal blood glucose level, no problem. But in diabetes, the blood glucose levels are so high that your reabsorption mechanisms are simply maxed out. And what happens is that the glucose transporters, they can't take care of all the glucose and reabsorb it, and so the glucose ends up staying through the loop of Henle distal convoluted tubule, and it ends up going out to the bladder with your urine. And then leaves your body. And so you can measure the glucose then in the urine, and that would be called glucoseuria. And then these dipsticks, they will detect measurable amounts of glucose in the urine, and that would be a strong indication that you have way too much glucose in the blood, which is called hyperglycemia. So way too much glucose in the blood results in glucose that spills into the urine, and it's going to be measurable, and it's usually an indication that somebody has diabetes. So that's the usual conclusion. Now, proteinuria would mean that you have protein in the urine, and that's not good. Typically, you might have a protein in the urine when the filtration membranes here in the glomerulus are way too leaky, and they're letting out the protein, which it should stay back so that you have your colloid osmotic pressure that you need in order to recapture fluids in capillary beds, such as this one here. And so sometimes people have what's called glomerulonephritis, where these filtration membranes are way too leaky and they're letting out the protein. You have zero reabsorption mechanisms for protein because it's not supposed to be in the tubular volume right here. And so every bit that got filtered out here, the glomerulus would end up in the bladder and then therefore in your urine sample. So if you're measuring protein in significant amount in the urine, that's not good. Moving on to ketones. Ketones are the byproduct of fatty acid metabolism, so beta oxidation cells. Now, if somebody's diabetic and they can't move glucose into muscle cells, for example, then the muscle cell has no choice but to metabolize fatty acids, and the result is a load of keto bodies that are being excreted in the urine. So if somebody has uncontrolled diabetes, you would find glucose in the urine, and you would find also ketones in the urine, and it gives the urine sort of this acetone, kind of fruity kind of odor. And people in ketoacidosis, they will also hyperventilate because these keto bodies, they add to the acidity. It's called ketoacidosis then. And so somebody with uncontrolled diabetes, you would find glucose in the urine, you will find ketones in the urine. You'll also find that they're breathing very heavily because they're trying to compensate for the extra hydrogen ions by breathing out more CO2. So and then their breath will smell like these keto bodies in the urine too. So that's the story on that. Now, we have a few more terms here. Urobilinogen, that is a substance that's the result of the breakdown and recycling of your hemoglobin, the red blood cells. Red blood cells only last for about 100 days, and they need to be recycled, and you're trying to recycle as much of the iron that was in the hemoglobin as well. Anyway, so the liver goes through processing. Whatever is water soluble will be excreted as urobilinogen with the kidneys. So it is normal to have a certain amount of urobilinogen in your urine sample. What's not normal is the bilirubin. It's also a byproduct of the recycling of red blood cells, but that's the portion that should be handled through your fecal material and be excreted that way. If you, for some reason, your liver doesn't work right or something else is wrong, then if you end up with bilirubin in the urine, that's not good, and it will make your urine sample more like a brownish, very dark yellow, brown basically. So that's not a good sign. A diuretic is any substance that will increase the amount of urine that you're producing, so it excretes extra water. Urea is a normal byproduct. It's a waste product, but it's also useful for recapturing of fluids for reabsorption purposes. Creatine is also a substance that will be excreted with the urine. A certain amount is normal, and it's actually used sometimes to test whether somebody's submitting a fake urine sample, maybe. And ketone bodies, we already talked about this up here. Those would be these guys here. They're typically found when somebody has diabetes uncontrolled, and the keto bodies end up in the urine because the poor muscle cells, they have no choice but to just exclusively process fatty acids for energy metabolism. Now, you might wonder, these keto diets, whether they would produce keto bodies in the urine. They might, very small amounts though, because you still have, if you're not diabetic, you still have insulin, and you will still have some glucose in the blood. It's normal, and so you can't really force your cells to exclusively process fatty acids only that way. Okay, so let's scroll down this lab, and you should have that at home with you. You can read through all of this. It's pretty much these pages are explaining what I just talked about, and the front page with the important terms. This page is kind of important. This tells you whether something is normal to have in the urine or not. So this is sort of your reference page to look up whether leukocytes, nitrate, etc. would be normal to have. And these are all the things that are tested on these chem strips. And you can scroll to all of this. The sediments, the urine sediments, we haven't been doing that experiment lately in the lab, so it's kind of hard to interpret a urine sediment. Anyway, so here are your general safety precautions, and then here the explanation of the procedures. The first experiment would have been the standard urinalysis, where you grab a pee cup and then you go to the bathroom and you pee into the cup, and then we do a chem strip analysis. And then we're going to take a look at what the expected result is just in a sec. And here, then the urine sediment, as I said, we usually keep that optional. Some people like to do it and take a look at their urine sediment under the microscope, but it's usually optional. And then experiment B is taking a look at unknown urine samples that you would have been given, and you would have just put the dipsticks into these unknown urine samples and found whether there's glucose in there or protein or some other substances that shouldn't be there, and sort of do a preliminary analysis of that. So the actual lab, the data report starts right here, and so we can sort of do a sample of this. Of course, you fill this out yourself, and the urine volume, whatever you brought back from the bathroom, the color should be some shade of yellow, depending on how your specific gravities, how many metabolites you have in your urine, either lighter yellow or even bright yellow or kind of more darker yellow. So there's a certain amount of cloudiness is normal. There's always some cells that are washing on as the urine leaves your body. It takes some of the normal microbiota with it and some cells from the bladder as well and the urethra. I guess you lucked out. You don't have to sniff it, because in the lab I would have made you take a whiff at your urine sample. Most people with a mixed diet have a certain amount of ammonia in their sample, and you can smell that. Also, you can smell coffee or asparagus and other metabolites. Now, the results from the chem strip. So, expected results. You should not have leukocytes, white blood cells, in measurable amounts. A trace amount is okay. Nitrate depends on your diet a little bit, but you should not, if any, it should be very little. It's sometimes used as a preservative in like beef jerky and stuff like that. Urobilinogen, a certain amount is normal. Protein should not be detected in appreciable amounts in your urine sample, and the pH should be somewhere between 5, could be as low as 5, could be as high as 8, depending on your diet. If you're strict vegetarian or vegan, then it's usually higher. Red blood cells only in very insignificant amounts. Blood is not normal to have in the urine. Specific gravity, depending on how much water you drink, your hydration status should be somewhere between 1.000, which is water, and 1.02, maybe on the high end. Ketones should not be in appreciable amounts in your urine sample. Bilirubin should not be in your urine sample. Glucose should also not be found in your urine sample. If you want to have a reference, or you're not sure about whether something is normal to be found in urine or not, you can always go back to this reference page right here about chemical characteristics. It will spell and explain each of the items to you. I'm back here on the data reports part of the lab. Part 3 would have been the specific gravity. We usually do this just with the chem strip. The urinometer is kind of a mess, and the refractometer too.